Okay, well, yeah, thanks, Angela, and thanks, Apala, for inviting me to this meeting, inviting me to give a talk. Really nice to be here. I've really enjoyed the last few days talking with everyone. So, so before I actually get started on the actual talk, I want to thank my collaborators. So most importantly, I want to thank Don Brass, Ben Fellows, and David Ewing. They're all my former and current grad students, and so without them, this work wouldn't exist. So they've done the lion's share of the work here. So I want to make sure I thank them from the outset. And then I'd also like to thank my ecology collaborators, Beth Purse, Amanda Callahan, and Stephen White as well. So the little video that you saw playing in the background, if you're wondering what that was, that's actually, let me start it again so you can see again. This is mosquito larvae. So these little things swimming around, they're the larvae. Those things that are static on the top of the surface of the water are pupae, mosquito pupae. So that's what's happening. You never really see them often, you just see the ones that bite you, but that's what they do at an earlier stage in their life cycle. So I'll be talking about them, but I'll talk about other insects as well. And I'll explain what we want to try and do is think about trait variation. So I will tell you what that is in a minute. In a nutshell, what I'm kind of interested in is how individual variations, so little variations from individual to individual, might interact at a population level to change the dynamics, and vice versa. The population dynamics might then shape the individuals you have in your population. And so fundamentally, I'm interested in that interaction and what that means for the population dynamics. So, so let me first of all then say, well, why insects? So let me tell you about why I'm interested in insects. So they're actually fundamental to how our ecosystems work. Let me give you some examples. I worked out I'd worked in all of these areas apart from one, so I'm working on that. So this up here, this is a bee, so they're important for pollination services, so transporting the pollen between flowers and ensuring that we basically have the food that we eat. Then we've got things like mosquitoes, so they're what we call vectors for disease. So mosquitoes, midges, for example, ticks, all examples of insects that transmit the disease, sometimes to humans, sometimes to animals, sometimes both. So again, a big problem for us with this disease transmission. Sometimes vectoring disease can be a good thing because sometimes you want to use them as to control other species. So sometimes you want them to vector disease. So it's not always bad, some of these things. On the right here is an example of forest defoliation by this guy here. This is called the spongy moth, and so these are oak trees, and it's basically when you have an outbreak of these, they just clear all the leaves off of the trees. And so this has implications then when we're thinking about carbon sequestration for forests, because obviously if they've not got those green leaves, then they're not producing the oxygen that we normally want. And so then they're going to have an impact on our environment that way. Also, there's other insects, for example, bark beetles, which also defoliate trees. And well, they kill the trees and lead to this kind of picture. But in that example, then sometimes you actually want it to happen, because what will happen with those systems is you'll get forest fires, and you might think forest fires are a bad thing, but actually they are a natural part of the ecosystem. And so for some species of conifer, for example, you need the heat of the fire to open up the pine cones and release the seeds, in those pine cones. And so without that, you wouldn't get the next generation of trees. So again, like a lot of these insects have good and bad aspects to what they do in our ecosystem. Then we've got their role in generally the food web here. You know, they're linked to everything in our ecosystem, and so they're important for our biodiversity. And then of course we've got like things like the locusts here, which are like in East Africa, where they're a real agricultural pest that we're concerned about. So they're important in lots of systems. And They're also very sensitive to the environment. So insects are what we call ectotherms. So what that means, essentially, you can think cold-blooded. So basically, they're not generating their own heat. It all depends on what environment they're in. So they're very, very sensitive to the environment they're in because of that. And so this little picture here, this flowchart, this is broadly speaking the life cycle of most insects. There's a lot more complications and nuances, but roughly speaking, this is how an insect's life cycle works. And so we've got eggs, larvae. So you saw some mosquito larvae at the beginning there, but also you think caterpillars, that's an example of a larvae, okay? So we go through this larval stage. Often there's several of these stages. They get bigger and they kind of get rid of their skin in between stages. And then eventually they pupate, so they're often like a little cocoon in that stage, and then become adults. So for example, like your flying mosquito here, or think butterfly, for example. And so often the adult stages might be the stages that do the most movement and disperse across the landscape. But all of these processes are dependent on the environment. They're highly dependent on the temperature. How quickly we move between the classes is very temperature dependent. They depend a lot on precipitation, and that affects things like survival and whether we're going to die as we move through these classes. Land use also affects these, and it can affect the level of competition, particularly what we see in the larval stages. It can affect the abundance of resource for these guys, again, which has an impact on competition. So they're very sensitive to the environment. So we've got insects, very important in our ecosystems. They're very insensitive to the environment. So then that begs the question of what does that mean if we think about climate change? So here's a little diagram here of some of the predictions of what climate change is potentially going to do to our environmental variables over the next century. So these colours are comparing our current state to what we expect to see at the end of the century based on sort of worst case scenario projections at the moment. And so on the right, for example, we've got you can see the change in temperature. So the top there's min and max. And you can see that, as we've sort of heard in the news, we're expecting things to warm. We're also expecting growing season to increase in some regions, but not others. More frost days in some regions, longer summers in some regions. And so we're really seeing lots of shifts, like more wet days in some areas, less in others. And so all of that is going to impact on what these insects do. So we really want to get an understanding of this. And we're already seeing these effects. So here's on the right here is an example of what those impacts are and what we're currently seeing. So because of warming and changes in seasonality, we're seeing rain shifts. So what that means is because of the warming, a lot of insects are actually moving further north to track the climate and to be in habitats they're most optimized for. So we're seeing invasions of species we've not seen before. So for example, Europe has seen the mosquito species that transmits dengue. So we've had outbreaks of dengue in Italy, and that's something you normally might just associate with more tropical environments. But because those mosquitoes are tracking the climate and moving north, we're now seeing them in Europe. Okay, so that's happening in lots of other systems as well. We also get changes in phenology. So phenology, that means timing, basically. So when different things happen. And so one consequence of changing in timing that happens because of this is, for example, if you're a caterpillar that relies on eating leaves, but you don't emerge when the leaves on the trees come out, then you've got no food, right? Or similarly, you might time yourself with the trees, but then the birds that eat you don't hatch their eggs early enough so that they're ready when you're abundant when the larvae are abundant, then they've got no food. So changes in timing can suddenly put a whole ecosystem out of whack. Okay, and so has a big impact on species interactions. Extreme weather events that we've also been seeing and hearing about in the news, they have implications for the fitness of the mosquitoes. Sometimes and other insects, sometimes it can mean more of them, sometimes can mean less. So if we're thinking bees, it's impacting them, we're getting less. That's a problem. For other insects, we're getting more. That's a problem as well. So there's lots of issues that we're already seeing happening. And so we really want to better understand this and better be able to predict what we expect to happen. So as I said, what I'm interested in is trying to put sort of individual variation into some of these models of these systems. And so that individual variation comes in the form of what's called phenotypic plasticity. So I'm going to explain about what all this means and what a trait is and everything. So let's start with what phenotype means. So your phenotype is a collection of traits. So if I just take like humans, for example, so let me just give you an example. So two of my traits that I have are my eye color, which is brown, and my hair being curly. Okay, so those are two of the traits I have, and like other people in the room have different traits, different eye color, different types of hair. Okay, so those are examples of traits, and the collection of those traits is your phenotype. Okay. So insects have that too. They have different colors, different shapes, but it's not just those things, but it's also things like your fecundity, your ability to survive. Those are also examples of traits, and those are the ones that I'll be more concerned about here as well. And that collection of those things is your phenotype. Okay, so then what is phenotypic plasticity? So this is different from thinking about evolution, right? So what is amazing about insects is they've got a certain amount of ability to kind of buffer themselves against environmental change. Because if you think about it, they live in a change, their world changes all the time anyway, just through seasons, through year-to-year differences. So they have a certain amount of ability to cope with change, and that's phenotypic plasticity, that's what that is. And so basically the idea is that its ability for an individual's genotype, so think your collection of genes, so they're somehow fixed, right? That you inherit those, you're stuck with those. But what can happen is, depending on the environment you're in, what traits those genes produce can vary in time, like over the time that you live. Okay, and so here's an example of this. So it's an ability to respond to the environment almost instantaneously. Okay, so here's an example. So this one is I think is fab. So these are caterpillars. So this and this is both a caterpillar, the same species, the same year, but slightly different times in the year. Okay, which is wild. They look completely different. And that's because this species is responding to the season. So this is in the summer, and it's trying to look like a twig on an oak tree. Okay, this is it in spring, and it looks like a catkin on an oak tree. And so this is at the same species, but it can respond to the environment, and so it can change as it's developing through the life stages, change what it is in response to the environment, so that it's adapted to the environment it's in at that particular time. Okay, so there's one example. Here's another example. This is the same butterfly species. You might think that's got to be something different, surely. This is exactly the same species, just in different environments. This is in the wet season versus the dry season, and you can see it's bigger and it's different color. And so this is quite common. So if you're bigger, then chances are you're going to be fitter, survive better, and lay more eggs, for example. So yeah, these organisms have got an amazing ability to adapt to the environment in real time. Here on the bottom is a really nice example. So this column is the males, and this is the female grasshoppers. And the top four of these are in a poor environment. So it's quite dry, and the vegetation quality is quite poor. And then these bottom four are in when the rainfall is much higher and the vegetation's lush. And you can see a massive difference in the size and shape of these insects. If we look at the females, you see how they've got this long abdomen here. So that means they're going to be able to, their eggs are inside them before they lay. So they're going to be able to produce more eggs. So that means that in these two different environments, these guys are going to produce more eggs than these guys. They're bigger, they're also going to survive better. So all these things are going to have implications on our population dynamics. And these two populations, this guy and this guy, they're only 15 kilometers apart. So the same time, but just 15 kilometers apart. And it's just because the environment's a bit different there that you see completely different grasshoppers. So they really are quite amazing. And so I want to include this kind of variation in my models. Okay, and I want to understand what impact does this ability to kind of do some kind of adaptation in real time have for our population dynamics and our ability to kind of cope with changes in environment. Okay. So here's an overview. So I'm going to, I'll introduce you to the mathematical framework that we're going to use to try and model this. Then I'll give you an example of by an experiment by Nicholson, who looked at sheep blowfly, and so that's lab data. And I'll show you how we can kind of use this framework to explain some of his kind of counterintuitive experimental results. Then I'm going to look at how actually you get there's some interesting math that emerges as well. So you get some surprising mathematical results and dynamics that we don't see in kind of some of the standard models that we might use. And then lastly, I'm going to show how we can then take this and really go to town on the modeling and then actually do something to predict disease risk of dengue from mosquitoes. So that's the plan. So let me start by telling you about the mathematical framework. So let me give you a bit of an idea of what people currently do, so you can kind of see where it sits in the field. So this is the current approach really to how you model phenotypic plasticity and how you think about it in populations. So people tend to think about, basically they tend to have the view that what happens at the individual level, you can kind of translate that as to the population level. So what I mean by that is in the lab, people measure these things called reaction norms. And so basically they're different environments. So if we think temperature, they rear the insect, for example, at different temperatures, like 15 degrees, 20 degrees, and then they measure the trait. So that could be fecundity, number of eggs it produces, how long it lives, for example. So these are what we call reaction norms. So they measure those in the labs. And then these are used in a variety of different ways to then predict what might happen under climate change, or if we've got they're competing with other species and so forth. So here's different examples. So one example is you might then compare these reaction norms between two species. And so you can then say, oh, well, this species will do better in this environment, this will do better in this environment, and make predictions that way. Another example is to actually use these to predict how trait distributions will change. So what I mean by that is you could estimate a distribution of the traits in the population, so the probability, like different individuals will have a particular trait, and then you can then use this reaction norm then to predict how that distribution will then change between environments. Okay, and so people use it in that way. In terms of population dynamic models, it tends to be used as putting this information into your parameters in the model. So if you've got a population model and you've got a growth rate, for example, and you wanted to include some aspect of plasticity, then what you might do is use a reaction norm like this to say, right, well, my growth rate is changing with temperature, so I'll put a temperature dependent growth rate in there. I'll feed in some temperature data, and that growth rate will change in time in my model based on the temperature. So that's the kind of common things people do. And so they're all good and they work to some degree in some situations, but what they miss out is that actually this isn't a one-way interaction. It's much more complicated than that. There's interaction, right? So the trait and the environment response, so this reaction norm, yes, it affects the population dynamics, but that can then shape the trait distribution in your population, which can then go back to interact here. And so this, in fact, can be a loop. Okay, and so you might think, well, how does this go and change the reaction norm? It will if your environment is the amount of food you have, for example. So then your population dynamics, how many individuals there are, will change your environment, so potentially change this response. So really, we want to capture that interaction. And so that's what we're doing with that framework. Okay, so here on the so what we're doing is we're going to take our insect life cycle and we're going to partition it into the into life stages. So here I've just shown you, so that's what this going vertically down is. I've got little end stages there, and so I've taken the life cycle of the insect and split it into end stages. Okay, so that's kind of classically what people might do. As I showed you earlier, eggs, larvae, pupae, adults, example. And then you might want to have these rates, so how long you spend in each class, for example, to depend on temperature. You might want your mortality to depend on temperature. So that would be more classical approach to this. What we've done instead is we've kind of expanded in this direction. So what is going on here, these extra boxes, is that's tracking what traits they have. So for example, the individuals here might have a different fecundity and growth rate to the ones here. And that's because based on the environmental cues they've experienced, they will sort of follow a different path through their life cycle. So we're basically keeping track of their environmental history, what they've experienced, to work out, okay, when you emerge as an adult, like what are you going to be like based on what you've experienced? So that's the basic idea of the framework we were developing to try and track that history of experience. So there's this is a particular example of one of those frameworks, and this one is an example of delayed developmental plasticity. So so you know, how I choose to construct these boxes kind of tells me how I'm capturing the life history of the insect I'm interested in. But so let me just show you what's going on with this one. So what this delayed developmental plasticity is doing is that so my population here, what I've done, I haven't got lots of boxes at the top here. I've actually summed them all into one. And the reason why is because all the individuals here are the same. So early on in the life cycle, they might all be the same. And I'm not seeing any effect of that plasticity. Okay. But at a later, so eggs, for example, like you tend to, eggs don't kind of do a lot. So, but when you get to something like larvae, the effects start to kick in. And so the environment that you experience then when you go from eggs to larvae might start to then affect what you are like as an individual, and you might start to change your chance of survival and so forth. And at that point, we keep track of it. And so, but also that might be delayed, and that's what we care about in particular, is that what determines the traits you have might not be your experience of the environment right now, but a bit in the past. So for example, the experience that a lot of insects have as larvae often determines what they're like as adults. So think the grasshoppers. If your conditions were quite good as larvae, then that might mean you're likely to survive better as an adult because you might be bigger and you might lay more eggs. So often, and that's what we're interested in here, and that's what we're capturing, is this delayed effect that the environment experience here actually determines what happens to you down here. So that's what we're trying to capture in these models. And that's what's delayed developmental plasticity is. It's like a delayed response to the environment. And these are the equations that we work with. So here is, so it's the rate of change of our number of individuals in each of my class. So I is the life stage and J is the environmental class. So that's telling me what traits I have. And so basically the structure of these models is you have recruitment into the class. So that's the incoming arrows. We've got maturation out, and then we've got some death. Okay. Now those things can be quite complicated functions, and the maturation term, that is, if you look, it looks like the same as the recruitment term, but it's got this delay in. So what we want to incorporate is the fact that you might spend a certain amount of time in a life stage before you move on. And so that's very common with insects, and insects are that's often temperature dependent, how much time you spend in a class. So that's what that delay is doing. So these are actually delay differential equations that I'm working with. And so what this maturation term is, it's the recruitment in the past. So how many came in the past, and then this S is capturing the probability they actually survived that whole period, tau, to actually make it back out the class at the end. So that's the basic framework for writing down these models. There's an expression for the survival. This survival here is just linear. So we've just got some rate at which you die, and it might vary in time, but it's just a linear rate. You can do things more complicated than that, but just gives you a bit of an idea of how we can set these up. And then the other bit to sort of complete the model is you kind of need to know what's happening in stage 1, the births. And so there we've got the birth rates, how many individuals, how many eggs each individual is producing. And this bit here is tracking the environmental experience. So it's sort of tracking who went in which class. And so we've got a way of keeping track of that. So that's the basic structure. If you're interested in sort of know more about the equations, please grab me in coffee break. I'm happy to talk about that. Okay, so let's actually apply this to a system. Okay, so here's the lab data we wanted to start with. So this is this dates back to the 1950s. Nicholson did these amazing lab experiments. So he monitored the blowfly. That's the picture of it here. This is a Australian sheep blowfly that he had in the lab. And so what he did was he's tracking the population, and hang on. So he started the population at high food, so unlimited food, and then he reduced the food after, I think it's 610 days. He lowered the food. So and the dashed line here is the experimental data from that experiment. It was you know, amazing. He was just counting all the time, so it was really high resolution data, really good data. And so then the black line here, the solid black line, is the output of a model that doesn't include this individual variation, this phenotypic plasticity that I was talking about. And so what you can see is it actually doesn't do very well. I mean, it's really carefully parameterized, really carefully parameterized, but it doesn't capture the dynamics. So let's look at some things that are notable here. So one is that if I look at the original data, you see the dashed line here. I've got oscillations. That's fine. We expect, you know, we quite often see that in these insect kind of models. And what happens is the food gets decreased. They kind of dampen, they stabilize. Okay. So we go from strong oscillations to a bit, the dynamics are stabilizing. So that's one thing. So that's one thing the model doesn't capture. The other thing that is probably surprising to a lot of you, unless you've seen this kind of thing before, is look at the population density. High food over here, quite low numbers, lots of food, higher numbers. So less food, more individuals. Right, that's not what we expect, right? And that's not what a classic model would produce, right? A classic model would say less food, less individuals, right? Kind of think that makes sense. So Nicholson had a theory. Nicholson had a hypothesis that this was due to phenotypic plasticity. So we thought, well, we've got a way of modeling this. So let's see what we end up with. Here's to kind of show that we really do have phenotypic plasticity in this system. So these are the reaction norms for this system. So here, if you look at the environment on these reaction norms is the amount of food each larvae have. So we're fixing the amount of food the larvae have in the system. We always give them the same amount of food. It's the adult food I was changing in that experiment. If we fix the amount of larval food, then what is going to affect the food per larvae is the number of larvae. Okay, so somehow the density of larvae is basically acting a bit like as a proxy for our environment here. And what happens is that the fecundity of the adults changes depending on the food the larvae have had. So this is an example of delayed plasticity. And then also the survival between getting from larvae to all the way to adults, that's also affected by the amount of food the larvae had. So this is an example of delayed plasticity, and these are the reaction norms. So these are the things that go into the models. So this isn't the model yet. This is just what's been measured in the lab. But what you can see is that the experience of a larvae affects how many eggs you produce and how likely you are to actually get to an adult, survive that far. So then we put that into our framework. So So I don't have to actually, so this is really a system that has eggs and pupae in as well, but I don't have to actually track those explicitly because not much happens. They just have to survive. So I can just incorporate that in without actually having to track the populations explicitly. So I can just think about larvae and adults. And so I've got my larvae. Larvae are pretty much all the same, but depending on the environment I experienced as a larvae, so how much food I had per larvae, that's where the reaction norms come in. That'll dictate which adult class I go in. It dictates what my survival was like and what my fecundity is like, my number of eggs I'm producing. Okay, so the environment that I get here dictates then which class I go into. So that's the model. And so each box here will have a different fecundity, so sort of eggs you produce, and a different chance that they survived from made it from larvae through the pupal stage to get to an adult. So they'll have different parameters, basically. That's the way to think about these. Each box here is a different parameter, different phenotype. Those parameters are your traits. Okay, so that's the setup. It's the same kind of equations that I showed you before. A few more details. There's our equation for the food. So that's this fixed amount of food the larvae have, but then I divide it by the number of larvae, and that's changing in time. So the food per larvae is really dictated by the number of larvae. Okay, so that's the framework. So then we looked at what happens when we simulate this. And so there's the dashed line is Nicholson's lab experiments, this data, and we do a much better job of capturing it. We've captured this stuff at the start really well. So this was when it was abundant food. And we do pretty good actually for here as well. Bearing in mind, we can't capture exactly what he did with the experiments in the models. It's a little bit tricky to kind of mimic what he's doing, but we do a pretty good job. Importantly, we capture the fact that you decrease food and the population goes up. We also capture the fact that those population cycles are starting to stabilize. So we've captured all of that by adding that detail in. And then these coloured lines here are what's going on with the individuals. So each colour, I'm just showing you the trait that's the adult fecundity, so the eggs are producing, and each colour is corresponding to a different level of fecundity, so a different one of those boxes. And so you can see that actually one of the things that happens is the composition of my population is changing as I change the food. Okay, so down here I've got a lot more yellow individuals with low fecundity, and then here I've got lots with the higher fecundity. So actually that trait structure is then really driving those dynamics. I'm changing what that population's composed of, those individuals. Okay. So that's good. So we're able to kind of replicate some lab data, and we're kind of able to explain some experimental results that couldn't be explained before. So what about is there interesting math that comes along with that? So we thought, well, let's have a look a bit more about what that model does. Let's explore the parameter space a little bit more. Let's have a think about this. And so that's what we did. And so here I'm varying the amount of food we give the adults. Okay. And so then what I'm seeing is I actually end up with, and then I've got the population density, larval and adults on the y-axis there. So these are bifurcation diagrams, basically. And so what you see, if you've come across hysteresis before, this is what we have. We have hysteresis. So once we explore parameter space a bit more, we found we've got hysteresis in the system. So what's going on here? So if you've forgotten what hysteresis is, or you haven't seen it before, then basically what's happened here, you typically have a saddle node bifurcation here and up here, and usually this is a stable solution branch, and then you have an unstable one here. That's typically what's going on. And so what's interesting about hysteresis is the following. So let's just look at this picture. As I'm changing my food, there's the environmental conditions. So changing a parameter, if I think about bifurcations, changing a parameter in my system. Then as I change the food, increase the food, the number of larvae go up, right? Oh, that's fine. That's what you expect. But what happens because you have these saddle node bifurcations, then this state suddenly the stable state stops existing. We've got an unstable branch here. And so the only option for the dynamics is to jump up suddenly to this stable branch on the top here. So this is what hysteresis does. And so from an ecology point of view, we often call these tipping points, where a small change in the environment leads to a large shift in the ecosystem. Okay, large shift in the dynamics. And so this picture here is what you would expect. So this is for the blowfly system, right? This is what you expect, right? Oh, changing food, increase food, I get more larvae. That makes sense. But then let's look at the corresponding picture. This is exactly the same. Everything's the same between these, but I'm just now putting adults on the axes instead of the larvae. So the total adults. And the S shapes flipped around. Okay, and so what's happening here is if I track, I increase food. So I could start off on this high branch, and then suddenly for high food, suddenly the population crashes. And the big thing with hysteresis, if I want to recover again, I can't just go back to where I was before in terms of food. Okay, I'm stuck on this stable branch now. If I want to go back, I have to kind of decrease food loads before my population goes up. That's just weird, right? Like, it's like, why on earth should decreasing food, and I need to decrease it a lot to make my population go up. So we ended up with these interesting dynamics that we were trying to understand. I'll give you a bit of an intuition for what's going on. So here's what's going on, and it's because of this delayed plasticity. So let's do a thought experiment. So I decrease my food. What does that mean? Less food as an adult, I'm going to lay less eggs. Fine. Okay, so less eggs, there's going to be less larvae around, so less competition. If I have less competition for food, then because of this delayed thing, the amount of food I have per larvae affects me as what I'm like as an adult. I'm going to be a bigger adult, so I'll have lower mortality to get to the adults, lower pupil mortality, and I will have better survival, and I will have higher fecundity. So more adults are reaching more blowflies are reaching adult, but that's a result of decreasing food. So so that's what's going on. It's this delayed effect. It's because although you might think lots of food is good, the trouble is it causes lots of competition earlier, and that's what dictates what the adults do. So so what we find is it's this incorporating this individual variation is really having an impact on the dynamics we expect. And we're actually be able to like pin down a lot of this analytically. So what these like solid lines here, the sort of horizontal solid lines, that's actually analytical results. So we're actually able to do a stability analysis of that model by hand, which was great, and in addition to the numerical results, which is what in the blue and the green there. So, and because we could get a handle on it analytically, we were able to actually say quite a lot. So these pictures here, these are the reaction norms I showed you earlier on this system. So where we had environment, food per larvae, and then how it affects fecundity and survival. But what we did was we thought, well, let's vary those, right? Because they're fitted to experimental data. There's a bit of uncertainty in the fits and so forth. There's some choice about what functions you fit. So we thought, well, let's play around. We can do that. And what we were able to show analytically is that regardless of what the fecundity did, the survival was really important. And we found that we needed to have the shape of this really mattered, and we could show it had to have a particular shape. In particular, had to almost have this S shape where it has low survival at low food and like a high survival, so almost like an S shape through here, what was necessary. And what I'm showing you here in the red and the blue, the red are examples of reaction norms that would lead to this hysteresis in the population dynamics, and the blue you wouldn't. So now we can start to say, oh, well, when would we expect to see this kind of behavior and these ecological, what we call tipping points, these crashes or big population jumps in real systems based on what's going on here. So we could actually use, we were getting interesting mathematical dynamics, and we could actually kind of talk, use that to kind of talk about things in general. There are lots of other types of plasticity. One is called maternal effects. So it's like what your experience as an adult affects what your offspring are going to do. We haven't looked at that. We want to think about that. There are lots more complicated life cycles than the ones I've shown you. But now we've got a framework for thinking about them, and we've got some math tools for studying them. There's some potentially interesting dynamics to start delving into. So that's one of the things that we want to do. Okay. So that's where we're going with the more theoretical side of things. But I want to sort of finish by showing you an application to a real world system. So I want to show you an application to dengue dynamics. So this graph on the right is showing you how vector-borne diseases, so diseases transmitted by insects, is on the rise. Diseases incident generally is on the rise, but in particular, the white there is the vector-borne disease. So more of it's been attributed to insects passing on disease. On the bottom here is the number of cases of dengue, and that's also been on the rise in recent years. And so to some degree, a certain amount of that is believed to be related to the effects of climate change. And so we were interested in trying to use our approach to look at this, because we know the traits in mosquitoes matter. Biologists are already aware of that the size of the mosquito, the wing size, can be quite important in what the mosquitoes are doing. So we thought, well, we've got an approach, let's have a look at this. So to give you an idea of the scale, there was over 40,000 deaths from dengue in 2017. In 2014, there was a big outbreak in Guangzhou in China. There was 14,000 hospitalizations. The outbreak lasted 7 months. And this was the first time people got interested in the mosquito we were studying. It's called Aedes albopictus, because up until that point, it wasn't thought to be a big player in dengue. But now suddenly, with this outbreak in Guangzhou, they suddenly realized it was. But what's key with this mosquito is it's invasive, and it's been able to adapt to kind of more temperate environments, and that's the one that's caused the outbreak we saw in Italy, for example. So Italy had an outbreak this year even. So and it's an invasive, and it's traveling north. So suddenly this is one we're worried about. And so what does this mean for us? Is dengue in Europe? Is this how common is this going to be in the future? So we used our framework. So this is, as you can see already, this is actually a more complicated life cycle than ones I've been showing. The eggs do various things related to weather, on precipitation and temperature. So there's a lot more biology going on here than I've shown in the previous models. But we can model it all, we can parameterize it all. And so we've got the effects of temperature affecting various stages. The key thing in here is that, as with the previous example, the amount of competition, so sort of food and resources that the larvae have dictate what type of adults they are. So it affects the wing length, which has an implication for survival and fecundity of the adult mosquitoes. So that's the key thing to take here, is that the way the plasticity happens, it's this delayed developmental plasticity again, and the environment of the larvae determines the type of adult that you end up being. So we put all that into our model. We managed to parameterize it all. And so, and this is an example of the climatic data that we got. So that's input in the model. And then the output is population dynamics, which is on the bottom here, over time. And then our trait distribution. So this is over time. The red line is the mean trait. So here I'm just plotting mean wing length, which is a proxy for survival and fecundity of the adults. And then the blue shading is the distribution. So we can track the whole distribution of traits in the population. So we can do that in these models. That's the output. The only input is the environmental data. So we then checked, okay, if all we do is I take our model that's been parameterized with lab data, we input environmental data that we can get. Let's see how that compares to the abundance data we could find. And this is what we got. So these are, so we went global, so North America, so here we've got like China there, we've got Europe, And so we got as much data as we could find, and we just compared our model to the data. So we're not fitting here, we're literally just comparing, and you'll see that we've actually done a really good fit. Where we could, we got data for different life stages. So overposition, this is the eggs basically. So the yellowy orange is egg data. That tends to be quite good data, so we do better there. And we've also got some trait data, not a lot, because there's not a lot collected, but we actually are capturing the traits where we've got it. We can capture multi-year data pretty well as well. So we do a pretty good job at capturing this and globally. So we're capturing across the range of this mosquito, from tropical environments to very temperate environments. So we've got a model that was working well. We were able to kind of look under the hood and try and explain some of the dynamics. So that's one advantage of having these mechanistic models. So these are just two locations. So Cagnes-sur-Mer in France. So this is what we've got here then is the trait, so the wing length. So there's not a lot of variation over the season in wing length. And these are the number of adult mosquitoes. And what these other coloured lines are comparing our approach to approach where we just assume that there isn't that trait variation. Every individual's got some average, which is kind of more typical approach. And that approach doesn't do too badly, but it does overestimate the season length. And that's because it almost says some mosquitoes are better than they are by taking an average, when actually at the end of the season we actually expect some quite like low survival of the mosquitoes, and it's not being captured by some of these approaches that don't take the variation into account. And it becomes more clear when we look at locations like Guangzhou in China, where the dynamics aren't seasonal, so there's a lot more variation in wing length. And there, if you take this kind of average trait approach and don't really incorporate that individual variability that we've been thinking about, you can do things like here and predict. You see how like some of the models here are going to 0 here, so they're predicting that things don't overwinter when in fact they do. And so not including these kind of individual level dynamics means you can like miss some of these important aspects of the mosquito dynamics. And this, you know, capturing the overwintering is then important for disease and whether disease is going to make it from one year to the next. So these kind of details do start to matter. So I'm going to quickly just tell you what that, so we had a model of the mosquitoes that work. We could extend that to actually put disease in. So basically we're taking kind of, if you're familiar with disease models at all, and I guess probably most people kind of at least know a little bit now through COVID, like SIR models. So you split your population into whether they're susceptible, don't have the disease, or infected or not. So we can do that. So we did that with the mosquitoes. So we added in we split the adults into the ones that don't have the infection and the ones that do. And we also have a population for the humans as well. Okay, so the infected mosquitoes are biting the humans, and that's how the disease is being transmitted. So we extended our model to include that, and we could parameterize all that as well. And we compared it to outbreak data, and we did pretty good. But what we were able to do was then look at, can we predict where we get autochthonous transmission of the disease? So what does that mean? So in Europe, how you end up with dengue cases arising is because someone's arrived in the country with dengue, they've been bitten by a mosquito, and then it's been transmitted locally in the population. So that's what this means. Okay, that's what that means. So and so what we were able to do is able to predict that really well. So the high density colored here is indicating sites where we expect there's a high risk of getting dengue transmission locally in the community. On the right, this is maps of mosquito abundance, and what you can see is that classic approaches that kind of ignore this trait variation that we've been worrying about, they can't like home in enough on the locations where you expect the disease, and you tend to see maps more like this. And you can say, oh, well, it's more suitable here than here, but they're not often specific enough. But by thinking about the traits, we're really able to pinpoint exactly where outbreaks, those circles are are where we're predicting outbreaks and there are actually outbreaks. So so our approach has really been able to kind of do quite a lot in this direction. And we've also been able to understand what's causing that infection as well. So so here is us just zooming in and looking at what's happening in time for a particular location. So here's Kangsamer again and Guangzhou. So what we did was we looked at how much of the infection was coming from which mosquitoes. So the green is the wing length, the average wing length in the population, and then the orange is the wing length of individuals. So this is how like long-lived they are and fecund they are of the infective ones. And what we were finding was that in all the places where we had outbreaks, it was at the start of the outbreak, if there was a lot of large mosquitoes around, they were transmitting the bulk of infection. So they were usually responsible for getting that outbreak going. They would then die off later in the summer and it would be smaller individuals that would take over. But what we're finding was like 80% of the infection at the beginning of outbreaks was attributable to 20% of the population. It was these large individuals that were causing it. So we were able to kind of use the model to kind of pick that out of the dynamics. So I'm going to wrap up then. So so we've been able to show how actually modeling this interaction between traits, population, environment, including those feedbacks, enabled us to explain some population dynamics that we couldn't previously explain. We also gave rise to interesting like mathematical questions, interesting dynamics that we want to study further. And we're actually able to use this vector trait like approach to be able to explain, you know, the timing of dengue outbreaks and the magnitude of locations. So one area we want to take this in the future is now start looking at climate projections and looking at, okay, well, can we identify potential hotspots for the future, and where do we need to be concerned? Where do we need to put our monitoring efforts and so forth? Okay, right, and I will finish there. Thank you.